Hi everyone, I'm Charles Gadd and I'm a postdoctoral researcher from Oxford University. I'm here today to present my work on multiscale analysis of somatic copy number alterations. And so, briefly, somatic copy number alterations quantify the change in copies of DNA segments. And these changes can occur across many scales. So we have at the courses scale changes that may affect the entire genome through whole genome doubling, to scales events that may occur only in small fraction of the genome with vocal aberrations. A multiscale analysis attempts to identify the events at each of these scales and the interactions between them. So on the left, we can have learner representation based in the top left of the figure based on the coarsest of scales, and as we go down to the bottom right, we can learn representations based on small focal events. And this is important because learning these types of representations may lead us to driver mutation, discovery of driver mutations, biomarker discovery, tumor evolutionary dynamics, or to detect intratumor heterogeneity. So we introduce a method called Wave LSTM. Wave LSTM is a wavelet-based long short-term memory encoder architecture, and Wave LSTM learns separate representations for each scale. It combines each of these representations using a self-attention mechanism into a single attentive, multi-scale attentive embedding, and we demonstrate Wave LSTM on two tasks. The first is the task of identifying clonal structures, and we demonstrate that Wave LSTM is able to recover the clonal structures obtained through state-of-the-art bioinformatics approaches, but on top of this, it's also able to discover novel subclones. Secondly, we demonstrate Wave LSTM on the task of predicting survival outcomes in a cohort of cancer patients, and we demonstrate that these representations are useful in improving survival outcome prediction. The preprint of the paper is already available, and you can find it using this QR code. And if you have any questions, feel free to talk to me at post ID 28.
Okay. Hello, everyone. I'm Remy Liu. I'm very excited for this opportunity to briefly advertise our work, context-specific network embeddings via contextualized graph attention. This is a joint work with Hao Yuan, Kayla Johnson, and Arjun Krishnan. There are a lot of human diseases, and many of them can be explained by the dysfunctioning of one or several proteins in the human genome. And several previous work, including ours, have shown that using protein interaction network embeddings is actually a pretty effective way of uncovering proteins that are associated with a particular disease. However, there is still a lot unknown about how the protein association works under different cellular contexts, such as tissue and cell types. And in this work, we aim to shed light on this problem of protein disease associations in different contexts. The idea of our approach is very straightforward. Instead of preserving the network topology of the context-naive protein interaction networks, we want to preserve the context-specific protein interaction subnetworks. For example, here are different subnetworks associated for these different contexts. And to achieve this, we designed a method called CONE, the idea that effectively and efficiently achieve this goal of preserving the network topology of different contexts. We do this by using a shared graph attention neural network that can be contextualized via a learnable context-specific embeddings. The training goal is to preserve the network topology of these context-specific networks. So to quickly show you some key results, in fact, we are able to achieve better prediction for disease-associated proteins when compared to when using context-naive method. And furthermore, our methods can shed light onto what are the biological contexts that are relevant for particular diseases. Here are just some simple examples. I'm happy to tell you more about it when you're in the poster session. So for example, familial bicuspid aortic valve, we found that using the CONE method, we found that heart is one of the top associated contexts, which is pretty naive. On the other hand, we also found some very interesting cases. For example, the hypochromic microcytic anemia. The method found out that small intestine could be one of the most relevant contexts, and we found out that this is also really reasonable. So if you're interested, come talk to us in poster number 12.
Hi everyone, I'm Wojtek, and my poster is about quick binds, a lightweight and interpretable molecular docking model that we developed in the group of Professor Carnegie at Columbia University. Molecular docking is a computational technique that predicts the structure of the ligand bound to its protein target. It's oftentimes used in early-stage drug discovery to optimize the drug affinity. The affinity of a drug candidate to its protein target based on the structure of the complex. And in principle, the structure can also be determined experimentally, but that's much more expensive and much more complicated than doing that on a computer. And if docking is supplemented by an estimate of the binding affinity, it can also be used to virtually screen large compound spaces for drug candidates, thereby further reducing the cost on increasing the speed of drug discovery. There are traditional methods, there are methods for this already out there, but the problem with current methods is that they achieve good predictions but at the cost of long runtimes, which in the case of physics-based methods is caused by sampling many different binding locations, many different binding poses, and in the case of recent machine learning models by increasingly larger models and shifting towards generative modeling and co-folding approaches, so predicting the structure of both the protein and the ligand from scratch. Instead, we developed quick binds, which is a lightweight method for rigid blind docking aimed at virtual screening applications by trading accuracy for speed. In terms of architecture, it's based on the AlphaFold2 architecture, which also makes it very topical based on the recent updates to AlphaFold2 for co-folding AlphaFold3. We benchmark quick binds on two data sets, the PD bind test set and the postbusters benchmark. These are plots of the success rate, so the fraction of correctly predicted complexes versus model runtime, either explicitly or by sorting the models according to their runtime. And we can see that, as intended, quick bind is orders of magnitude faster than traditional docking methods, than the current state-of-the-art docking method, DiffDock, and recent co-folding tools. But it still outperforms most other docking methods except for DiffDock and co-folding methods such as AlphaFold3. Given that a natural use case for a quick bind is virtual screening, so we developed a simple top model for binding affinity prediction such that it can be immediately used for virtual screening applications. And that's particularly important because most other docking and co-folding tools do not actually predict the binding affinity, so it cannot be used for virtual screening at all. So in a nutshell, a quick bind optimizes runtime speed while retaining capacitive accuracy and is therefore a compelling option for high-throughput virtual screening. And if you want to learn more about quick bind's architecture, performance, model interpretability, or customizability, come to my poster.
Hi, my name is Masatoshi. Today, the goal of this work is a computational design, like for biological sequences. Especially, we focus on bridging model-based optimization and also generative modeling. So what is model-based optimization? It's a very simple approach. So first, like we make a sequence model from X to Y, for example, protein to like the affinity, or like DNA to like cell type specificity. And then we can optimize that with directed evolution or like the first order method. So that is a traditional approach. And another recent popular approach is also generating modeling approach. So we make a language model or diffusion model for sequences, and then like we can generate basic sequences. So the question here is that the world model, so model-based optimization, it excels in generating sequences with high functionality, and also generative modeling excels in generating sequences with more natural sequences. So here, the goal is to construct a new method that can enjoy the best of both worlds. And to achieve that, we propose a new RL-based fine-tuning approach for biological sequences. So here, let's assume that we already have a good generative model trained on large data sets. And then also like we have access to some reward model from sequence to functionality. And in our approach, gradually, like interacting with reward model, we fine-tune like generative model using reinforcement learning. And then, for example, we can generate like DNA sequences with high serotype specificity. And there are several challenges. So, for example, like in this kind of stuff is researched a lot in computer vision. But compared to this literature, there are two challenges. And the first challenge is that since we are dealing with more discrete objective, like basically sequences, so how to incorporate this like discrete diffusion model in our context is a challenge. And another challenge is that the coverage of the functional data is very limited. So like we can easily generate basically out of distribution samples if we just use a naive approach. So in the interest of time, I don't talk about how we deal with these challenges. But if we are interested, let's chat at the poster session. Thank you very much.
Hi, everyone. I'm Sabina. I'm a postdoc at Dana-Farber, and I'm going to be presenting my poster, which is entitled Precision Prognostics in Pediatric Neuro-Oncology using DNA methylation arrays and neural networks. So to motivate this problem, I want to stress how important it is to accurately predict the outcome, especially in pediatric patients. This is because we'd like to manage the trade-off between undertreatment, which can result in recurrence as well as death, as well as overtreatment, which can result in lifelong debilitating side effects. So to address this problem, we've collected data from patients with medulloblastoma as well as ependymoma, and we have this DNA methylation array data, as well as the associated survival outcomes, which is sensor data. And from this data, we can also obtain copy number variations. So we feed this into a sparse neural network, which then produces patient-specific survival probabilities over the course of 10 years. So I mentioned the sparsity of the neural network, which also enables it to be more interpretable. So we can determine for each patient which genes were highly predictive of outcome. And this has enabled us to also uncover new biomarkers. And lastly, we adapted the strategy using a causal inference framework to estimate individual treatment effects from patients which were randomized within a clinical control trial. And we noticed that there was significant heterogeneity in treatment response, which would otherwise be missed when looking at aggregated data. If you're interested in talking more, I'm looking forward to seeing you at my poster.
Good day, everyone. I'll be presenting the last spotlight for today. I'm Oladimeji Macaulay. Oh, that's one more. Okay. I'm Oladimeji Macaulay, and I'll be presenting MedGravNet, which leverages multi-relational, GNN, and text knowledge for biomedical prediction. It's really important to understand the interactions between biological entities in order for you to understand the association of these factors to diseases. In order to address this, we introduced MedGravNet, which is a multi-relational graph neural network which infers relationship between 4 biological entities, which are gene, drug, phenotypes, that is the side effect, and the diseases. In order to compile the data, we got it from several databases. And for the model architecture, the nodes were initialized using embedding from a language model. And then we trained the GCM for the link prediction. MedGravNet was able to outperform single relational graph and multimodal fusion, which will be shown in the poster. Also, we also try to verify the generality of this model by implementing several assessment methods, which is trying to train on a set of disease gene association and then predict disease gene not included in the data set, that is the one from another database. Also, we also tried to see if we could predict risk genes for rare disease. And how did we model this? We modeled this by removing all known information of a particular disease from the graph, which is used to imitate a real-life scenario of a rare disease, and the model was able to perform well. Also, side effect prediction. We removed the side effect data entirely from the graph and tried to use the model to predict it, which the model performed very well. And for more information about this, I'll be at poster 18. Thank you.
Hi everyone, I'm Jessica. I'm a postdoc in the Q Lab at Cold Spring Harbor, and I'm here to plug our work addressing uncertainty in machine learning for biology. In recent years, deep neural networks have become a very popular and powerful tool for studying genomic sequence function, which is why we're all here today. But it's not always clear how well they generalize to out-of-distribution sequences, or DNA sequences that differ significantly from what the model saw during training. So it'd be nice if we had a way to quantify how certain our models are about their inferences, as this would give us a way to improve the quality and reliability of the biological conclusions that we draw from the application of these tools. One way to address this issue is with deep ensembles, which involve the use of multiple deep neural networks for a single prediction task. And typically, the predictions from the different models in the ensemble are combined to yield a single prediction, which has been shown to have improved performance over individual models. And additionally, the degree of disagreement across the individual predictions reflects the model's uncertainty for the given input. And this quantity represents epistemic uncertainty, which is one of the two primary sources of predictive uncertainty. Now, while deep ensembles offer notable advantages over individual models, they also suffer from several limitations arising from their dependence on multiple models. And so to address this, we leverage a method known as ensemble distribution distillation, wherein we essentially generate new target labels for the data using the ensemble's predictions, which are then used to train a distilled model that mitigates the disadvantages of deep ensembles while also preserving their benefits with just a single model. And more specifically, we achieve this by assigning the average and the standard deviation of the individual predictions from the models in the ensemble as new target labels for the data, which we then use to train a distilled model wherein the output layer is modified to include a new output head for epistemic uncertainty, which is learned from the ensemble standard deviation and trained alongside the original prediction task. And we applied this method to different genomic sequence models across various functional prediction tasks, and we were able to demonstrate that our distilled models do indeed yield improvements in performance over individual models, and additionally are able to predict epistemic uncertainty quite well. Having said that, epistemic uncertainty alone doesn't offer a comprehensive view of predictive uncertainty. Ensemble distillation doesn't capture aleatoric uncertainty, which arises from inherent noise or uncertainty in the data that the model learns from. So in the case of genomic sequence data, that could be like technical or genetic variation. So we estimated aleatoric uncertainty by training our models on the variability across biological replicates in the data, and then applying distillation to these models to yield uncertainty-aware models that can estimate both aleatoric and epistemic uncertainty. And when we evaluated these uncertainty estimates and compared them to those from several other popular methods for uncertainty quantification, we found that the total uncertainty estimated by our distilled models were quite well calibrated, and that we could additionally ensure further statistical guarantees when applying conformal prediction to these uncertainty estimates. So for more details on everything I've just shared in the Spotlight talk and more, make sure to check out my poster at this afternoon's poster session. I'm poster number 60. Thank you.
All right, that's the end of the Spotlight session. So now we have the poster session, which is in the Allen Atrium. So if you go downstairs, cross the little road, there's the Allen Center building, and then just go inside. You should be able to see the poster boards. And see you guys all there in 5 minutes.